Please give a nice welcome to the team from UC Davis.
Hello, we're Team Cenozoic from the University of California at Davis. This year we developed a mammalian cell-based bioassay which reports physiological stress for use in environmental toxicology. All the work for our project was done under a biological use authorization from our university. In the far northern end of California, a Native American tribe lives at the mouth of the Klamath River, where their people have lived for countless generations. This community exists in close relation with the local ecosystem, which they rely upon for food, materials for their traditional ceremonies, and recreation. Our team joined fellow researchers from our university to visit the tribe and learn more about their way of life, what they value, and how various activities impact their community. In recent years, the tribe has published elevated rates of cancer, miscarriage, and other illnesses relative to what they expected. Upriver from this community, industrial agriculture and forestry corporations manage the land. These corporations obtain control of this land through long-term contracts, which leaves the tribe with little power to control how the land is used. As part of their routine practices, these corporations apply herbicides, pesticides, and other industrial chemicals upon the land under their control. These companies claim to be following existing regulations regarding the use of these chemicals. However, these chemicals have been detected in the water of the river and in local flora and fauna. Additionally, illegal marijuana growers in the region have been found to be using warfarin, a potent rat poison, to protect their crops from pests. This chemical has also been measured in the environment. Copper compounds have also been detected in the environment in the Klamath River region. These compounds are naturally occurring due to the underlying geology of the region. However, the rate at which they are introduced into the environment is accelerated due to erosion caused by human activities such as logging. The tribe has presented the hypothesis that the trend in illnesses which they have noticed may be linked to the presence of these chemicals in the environment. After listening to the tribe discuss their hypothesis, we asked ourselves how synthetic biology could possibly be used to help develop a better understanding of the possible links between the presence of these chemicals in the environment and human health. As we went about designing our project, we took careful measures to integrate human practices every step of the way. And a key part of human practices, as we discovered, was paying careful attention to the precise cultural context in which any solution involving synthetic biology would be used. The tribe has previously voiced concerns about biotechnology. In 2015, the Tribal Leadership Council passed the Genetically Engineered Organism Ordinance, which made illegal the creation, growth, or release of genetically engineered organisms into their environment. This ban was passed in direct response to the FDA's approval of transgenic salmon, a species which has cultural significance to this community. Interestingly, when our team read through the law in question, we learned that the ban has very large exceptions. These exceptions allow for all of bacterial synthetic biology, as well as for genetic modification done for the purposes of education and biomedical research. When we reached out to a member of the tribe's scientific body for clarification, the tribe's scientists told us that while the introduction of transgenic organisms into their environment was incompatible with this community's values, other forms of biotechnology were not. One such acceptable use was as a tool for biomedical research in order to learn more about protecting human health. With a better understanding of the cultural context in hand, our team set out to help find a way to demonstrate a proof of concept by which synthetic biology could be used to help develop a better understanding of possible links between chemicals in the environment and human health. Moreover, we wanted to find a way to do this within the context of iGEM, which has limited time and resource constraints, and we wanted a demonstrable proof of concept by the end of the summer. Of all the possible projects in iGEM, some were immediately apparent would not be appropriate. One such inappropriate project would be release of transgenic organisms into the environment for in situ bioremediation. Another popular project in iGEM is the creation of a bioassay. Now, at this point in the conference, I'm sure you're all thinking, not another iGEM bioassay. Haven't we seen them all? Well, this one's different. Let's show you why. As you may know, an assay is a technique by which a chemical's potency, concentration, or effects are characterized by how living cells respond to it. In a typical assay, E. coli is engineered to report the presence or absence of a specific chemical of concern. But the tribe's concern was not about measuring the chemicals present in their environment. They already knew what chemicals were present. What they wanted to know was what effect they had on the health of people in their community. The answer wasn't a bioassay to detect a substance, but rather a bioassay to detect a phenomenon, the stress inflicted on mammalian cells. The gold standard for studying the physiological effects of chemicals is whole animal in vivo studies. However, these studies require substantial time, money, and resources. Additionally, these studies expose animal models to harm, and bioethical considerations require that we minimize animal suffering. Therefore, we determined that the appropriate chassis for a biosensor was mammalian cell lines. These cell lines are inexpensive compared to animal models, but retain the ability to link the presence of a substance to mammalian stress. When mammalian cells are exposed to a chemical which induces stress, they activate complex cellular pathways comprised of many genes. These pathways are comprised of so many genes and are so large and complex that it would be very difficult to engineer them into a simple host organism. And even if you could do that, there is almost no guarantee that they would behave in biologically relevant manners. So instead, we created a simple genetic construct which we introduced to mammalian cell lines, taking advantage of the already present genetic architecture. Before we designed our project, we decided that we wanted to set out some criteria for success. We decided that in order to assess our project as a successful attempt to help bridge this gap, we wanted to be able to measure several chemicals of concern present in the Klamath River Basin at or below concentrations already determined by the EPA to be relevant to human health. The rest of our project would follow a typical engineering design, build, test cycle, followed by a validation step in order to assess whether or not we'd met our goals. We began by selecting our chemicals of concern. For these chemicals, we selected chemicals which are representative of what is found in the Klamath River Basin. We then have researched which cellular stress pathways they've been associated with in the past. For our experiment, we selected stress pathways associated with oxidative, heavy metal, unfolded protein, and genotoxic stress. These stress pathways have been implicated in many diseases. We then isolated inducible promoters from genes in these pathways and connected them to a reporter gene, eGFP, which has been optimized for use in mammalian cells. We then assembled our constructs into plasmids and transfected them into mammalian cells using lipofectamine. For our experiment, we used the CHO DG44 cell line, which is derived from hamsters. This is a very commonly used cell line for pharmaceutical production and has been widely studied. It has also been used for toxicology studies in the past. We chose not to use human cells for our project due to the time and resource constraints of the iGEM competition, as well as to make our work more easily replicated by future iGEM teams.
So after inserting our promoters into our mammalian cells, we ran a series of initial experiments to optimize our seeding density, time before transfection, volume of lipofectamine, DNA amount for transfection, incubation time before induction, and optimal induction time. And just in case you weren't taking notes, all of these experiments, along with our protocols, can be found on our wiki. We tried to capture some of what we learned this summer into these protocols in hope that they may help future teams in iGEM get into the historically underdeveloped space of mammalian synbio. Initially, we measured the fluorescence intensity of our bioassay using a microplate reader, but we found that this method resulted in a high autofluorescence from the complete growth media. And in an attempt to combat this, we washed our cells with phosphate-buffered saline before testing at each time interval, but we found that the PBS wash decreased cell adherence. While we found an alternative media optimized to reduce autofluorescence, this product was back-ordered. We therefore chose to use a fluorescence microscope as our tool for measuring induction. Then we started testing. We ran 2 sets of experiments. In our first set of experiments, we tested all our promoters against 2 chemicals of concern, copper sulfate and hydrogen peroxide. And from this data, we found that our promoter metallothionein 1 showed the most promising induction curve, and therefore we optimized our measurement protocol to further characterize our MT1 promoter construct. For our second set of experiments, we tested our MT1 constructs against the complete panel of chemicals of concern. Measurements were made at 0, 12, and 18 hours post-induction. For each concentration, we collected 4 data points. And to ensure uniformity between the images, the camera settings were manually fixed to be the same for all images. Once all the pictures were taken, they were analyzed using ImageJ software. And we wrote a script that allowed us to analyze only the parts of the image that displayed fluorescence from the cells. And the parameter values used in the script were optimized to select areas of interest and reduce noise. By the end of our project, we had collected 20 gigabytes of data and processed over 2,500 images. So with all the images processed, we wanted to quantify our stress. Our data processing pipeline needed to control for differences in cell number between the wells, differences in transfection efficiency between the wells, and account for the noise from both extrinsic and intrinsic sources. To control the differences in cell number, we visually confirmed that each well was between 70 and 80% confluent prior to induction. And to control for transfection efficiency, we transfected our cells with a positive control in which CMV, a constitutive promoter, was coupled to eGFP. And we analyzed the variation between these positive control replicates by comparing the coefficients of variation. We found intraplate variation to be much lower than interplate variation. Therefore, we controlled for transfection efficiency on an intraplate basis. Next, we needed to account for intrinsic and extrinsic noise. We found that there was a baseline expression level associated with our uninduced promoters, which we then accounted for using our control samples. And then after adjusting for this intrinsic noise, we observed that glucose, our negative control, displayed a low level of induction. To account for this extrinsic noise likely introduced from our experimental procedures, we subtracted the glucose fluorescence values from our normalized fluorescence intensities. So to reiterate, equations 1 and 2 were used to normalize our data, and equations 3 and 4 account for intrinsic and extrinsic noise. Next, we generated induction curves by plotting the corrected normalized fluorescence intensities against the concentrations of chemicals of concern. Who are stressed? I am. Well, the goal of our project was to find stress using mammalian synthetic biology, and we did. Here, we see the corrected fluorescence values obtained from the induction of the metallothionein-1 construct by hydrogen peroxide. The x-axis shows the concentration of the chemical of concern, and the y-axis shows eGFP production in terms of fluorescence intensity. In this graph, we see the corrected fluorescence values obtained by the induction of our MT1 construct by copper sulfate. In this case, we see that the corrected fluorescence begins to decrease after about 1 micromolar of concentration of copper sulfate, and we hypothesize that this is due to the cells dying at higher concentrations of copper sulfate. And this phenomenon was also seen in our experiments using the chemical warfarin. Next, to quantitatively characterize our results, we took this data and fit it into a mathematical model. Our model is a derivation of the Hill equation commonly used to model a dosage response curve. And to this, we added an additional term that takes into account cell death. Next, we were able to determine from this data analysis the sensitivity of our promoter to our various chemicals. We repeated the same process for all of our chemicals of concern and one environmental sample. Next, we compared our results to literature and EPA-cited values for toxicity. Let's consider copper sulfate. The EPA tells us that the relevant cytotoxicological dose for copper sulfate corresponds to a concentration of 6.25 micromolars. And we found that our promoter reports a stress induced by copper sulfate at a concentration of 0.6 micromolars. This means that our promoter construct can sense a stress induced by copper sulfate at 10 times lower concentrations than the threshold defined in literature. If we take a look at warfarin, we can make similar conclusions. And while there's much more work to be done, these results suggest that we have met the conditions for success that we set out for ourselves in the beginning of summer. That is, to create a mammalian bioassay that can be used to detect cell stress for multiple different compounds found in clamates at or below the cytotoxic conditions. Yay. So, to contrast between bacterial and mammalian biosensors, we collaborated with the Leidens iGEM team. Hello. And the Leidens iGEM team were characterizing stress pathways as a means of augmenting antibiotic use. They showed that nalidixic acid, an antibiotic used to treat urinary tract infections, induces cellular stress in bacteria. They hypothesized that this chemical might also induce stress in mammalian systems and asked us to investigate using our bioassay. Here, we see the corrected fluorescence values obtained from the induction of the metallothionein-1 construct by nalidixic acid. Our data suggests that nalidixic acid produces a small but detectable induction of our promoter. So together, our data and Leiden's data suggest that nalidixic acid has the potential to cause cellular stress in both bacterial and mammalian systems. And this is a particularly interesting result considering the use of nalidixic acid in a pharmaceutical context. We look back on this collaboration as a very positive influence on our project as we learn how our constructs may be used in a pharmaceutical sample as well as an environmental sample. Thank you, iGEM Leiden. Through this journey, we became very interested in the specific sets of environmental, cultural, and legal contexts in the Klamath region. We consulted with experts in the Native American studies and members of the tribe in order to further investigate the relationship between indigenous communities and biotechnology. We compiled all of our findings into a comprehensive case study that can be downloaded from our wiki. And it's our hope that this document can aid future iGEM teams seeking to become more engaged with indigenous communities and other groups who have concerns about biotechnology. Our project will continue to live after iGEM through the Bioinnovation Group at UC Davis. This is an undergraduate-run research organization, and the students of this group will develop stable cell lines, test our construct in human cells, and screen more compounds against our bioassay. Additionally, we aim to explore the synergistic effects of chemicals of concern on our constructs to understand how these chemicals affect our mammalian cells. We're deeply grateful to our mentors and advisors for making this project possible, especially Dr. Mark Fajati and Andrew Yao. And we would also like to thank the experts we consulted in the fields of environmental toxicology and Native American studies. A special thank you to our designer, Linda Wong, for her support to this project. And we would like to thank our sponsors for supporting our research and making this project possible. And finally, we would like to thank you for listening.
Beautiful project, guys, beautiful presentation, and especially thankful for the documentation you prepared for future teams, both about the protocols and your integrated human practices. I think it's great for everyone in the future, and it's very important. And I wanted to ask about your lab results with Mark. Yeah, I particularly like that you tested antibiotic, a substance that's supposedly pharmaceutical, but it's also apparently poisonous to humans. So I think your assay has good potential. It's good work.
One of the key things that we learned from cancer biology is that findings in cell lines often do not translate to animal or human models. What was your thought process of that?
Thank you for that question. So we actually became aware of this whole question of bioavailability when we discussed this with an environmental toxicologist from our university. One example he gave was that many endocrine disruptors present in foods, if you expose them directly to cell lines, they cause tremendous damage, but they're so readily digested within organ systems that this isn't a problem at all. And we were definitely very interested in this question of bioavailability, and we didn't have time to directly address it within the course of the iGEM competition. But with our extensions through the Bioinnovation Group, we definitely want to be aware of how the cells behave differently within the context of an organ system or an organoid. And one of the side projects in this organization is actually a bioprinter, and they're interested in the possible applications of printing organoids in a three-dimensional conformation. Thank you for that.
Great, thank you.
Thank you. It was a great presentation and great project. So, how do you plan to use it in the field to detect? So, you have the bioassay working. How do you plan to use it, for example, in the Native American community?
So currently it's not robust enough because we're using mammalian systems, which tend to be much more sensitive to. So as a field test, it might not be feasible to use it in that context. But the most reliable way we would use it would probably be take an unknown sample from the environment and then expose it in a laboratory setting to our assay, and hopefully that gives us the result.
Another addition to that, the tribe has serious concerns about the contamination of their local ecosystem with transgenic organisms. And they've made it very clear that while there are certain sets of conditions in which they find them to be a valuable addition to human knowledge, they're really quite interested in the parameters and the control and the safety. So the tribe has indicated that they feel most comfortable with this being confined to a laboratory space.
Thank you. And can I continue? Following on that, you show that exactly that graph there, the promoter is sensitive also to not only to the copper sulfate that you showed first, but also to this one. So in the context of using it, for example, with soil from the community or something like that, how will you deal with the problem of specificity?
Yes, I can actually answer that one as well. So something that we were really interested in is there's already very good tools in environmental toxicology for detecting the presence or absence of specific compounds. These range from analytical chemistry, such as NMR and other test reactions, as well as antibody-based approaches, such as ELISAs. So we really view ours as operating within the space of you take a sample, and then you screen it against our assay, and you say, okay, something in this causes oxidative stress, and this is warrants further study. And then, because with a lot of these analytical chemical methods, you have to already know what you're looking for. So this can really help direct future study and can direct the attention of researchers to particular areas of interest.
Many of these toxic compounds only cause any effect after a long exposure time. Have you thought about that at all?
Yeah, so one thing that we're interested in is we selected stress pathways rather than, for example, cell death as our criteria, because it's very difficult to test with a lot of these things long-term exposures. And our hypothesis that was integral to our study was the idea that if you have even a very low dose, it's probably going to be causing some detectable activation of stress pathways. And like I said, many of these stress pathways have been implicated in human diseases. The data is currently still being gathered, and some of these claims are quite contentious. But there are researchers who feel that there are